fluorescent sensors, which can be used to label and image a wide variety of molecules, offer a unique glimpse inside living cells. However, they typically can only be used in cells grown in a lab dish or in tissues close to the surface of the body because their signal is lost when they are implanted too deeply. MIT engineers have now come up with a way to overcome that limitation. Using a novel photonic technique they developed for exciting any fluorescent sensor, they were able to dramatically improve the fluorescent signal. With this approach, the researchers showed they could implant sensors as deep as 5.5 centimeters in tissue and still get a strong signal. This kind of technology could enable fluorescent sensors to be used to track specific molecules inside the brain or other tissues deep within the body for medical diagnosis or monitoring drug effects. Scientists use many different kinds of fluorescent sensors, including quantum dots, carbon nanotubes, and fluorescent proteins to label molecules inside cells. These sensors' fluorescence can be seen by shining laser light on them. However, this doesn't work in thick, dense tissue or deep within tissue because tissue itself also emits some fluorescent light. This light, called autofluorescence, drowns out the signal coming from the sensor. To overcome this limitation, the MIT team came up with a way to modulate the frequency of the fluorescent light emitted by the sensor so that it can be more easily distinguished from the tissue autofluorescence. Their technique, which they call wavelength-induced frequency filtering (WIFF), uses 3 lasers to create a laser beam with an oscillating wavelength. When this oscillating beam is shined on the sensor, it causes the fluorescence emitted by the sensor to double its frequency. This allows the fluorescent signal to be easily picked out from the background autofluorescence. Using this system, the researchers were able to enhance the sensor's signal-to-noise ratio more than 50-fold. One possible application for this kind of sensing is to monitor the effectiveness of chemotherapy drugs. To demonstrate this potential, the researchers focused on glioblastoma, an aggressive type of brain cancer. Patients with this type of cancer usually undergo surgery to remove as much of the tumor as possible, then receive the chemotherapy drug temozolomide (TMZ) to try to eliminate any remaining cancer cells. This drug can have serious side effects and it doesn't work for all patients, so it would be helpful to have a way to easily monitor whether it is working or not. When temozolomide enters the body, it gets broken down into smaller compounds, including one known as AIC. The MIT team designed a sensor that could detect AIC and showed that they could implant it as deep as 5.5 centimeters within an animal brain. They were able to read the signal from the sensor even through the animal's skull. Such sensors could also be designed to detect molecular signatures of tumor cell death, such as reaction oxygen species. In addition to detecting TMZ activity, the researchers demonstrated that they could use WIFF to enhance the signal from a variety of other sensors, including carbon nanotube-based sensors that the team has previously developed to detect hydrogen peroxide, riboflavin, and ascorbic acid. The researcher says that the technique works at any wavelength and it can be used for any fluorescent sensor. For this study, the researchers used 3 lasers together to create the oscillating laser beam. But in future work, they hope to use a tunable laser to create the signal and improve the technique even further. This should become more feasible as the price of tunable lasers decreases and they become faster. To help make fluorescent sensor easier to use in human patients, the researchers are working on sensors that are biologically resorbable, so they would not need to be surgically removed.